Hello again, and I'd like to introduce our final panel of the afternoon. Platforms everywhere, how to differentiate your vaccine platform in a competitive market. Your moderator is Joshua Speidel, who's Managing Director, Commercial Practice Team of the Latham Biopharm Group. Please welcome Joshua.
All right, thanks everybody for sticking around for our panel session. This panel is focusing on how companies that are emerging with technology platforms, how they should market themselves, how they should think about it from a regulatory perspective, what are the challenges associated with being a manufacturing platform, and how some of these companies that are in our panel have addressed those issues. And so I'm Josh Speidel with the Latham Biopharm Group. We're a life sciences consulting firm, and we work large. We have a number of programs with the U.S. government developing platform technologies for biodefense and pandemic influenza. My first exposure with platforms came when I was working as a consultant to DARPA, and they were developing new manufacturing platforms for vaccines under the AMP program, which was focusing on developing 3 million doses in 12 weeks, and so they needed the efficiencies of a platform approach to the program. So I'm going to turn to the panelists and ask that they introduce themselves, but also introduce the technologies that they are working with now. So Bill, would you start it off?
Hi, I'm Bill Taylor. I'm Vice President of Business Development at Protalix Biotherapeutics. Protalix is an Israeli-based company that's developing treatments for orphan rare diseases. Right now we're focused on enzyme replacement therapies. We have a commercial product already in the marketplace, Elelyso, to treat Gaucher type 1, and a phase 3 program to treat Fabry disease. We utilize a novel plant cell culture-based technology platform. So we have the distinction of being the first and only FDA-approved product that's utilizing plant cells in suspension culture to produce a human therapeutic that's in the market today. Prior to my job at Protalix, I worked with the team at Phoenix, and Diane will introduce the Phoenix platforms, but I worked in the business development group during my time at Phoenix as well.
I'm Diane Retallack. I'm Senior Director of Upstream Processing and Intellectual Property at Phoenix. And we are a clinical stage biotech company focusing on biosimilars and novel vaccine development. And our platform technology is a Pseudomonas fluorescens-based expression system that we use to produce our biosimilar therapeutics, as well as developing novel vaccines such as the next generation anthrax vaccine.
Hello, my name is Rachel Felberbaum. I'm a Senior Director of Business Development for Protein Sciences Corporation. And we utilize the baculovirus insect cell platform to make recombinant proteins, predominantly focused on vaccines. We have one commercial product, which is called FluBlock. It was approved by the FDA in 2013 and in Mexico in 2014. It's a recombinant hemagglutinin protein, so a pure protein-based influenza vaccine. And importantly, we've shown in a large clinical study of adults 50 and over that the recombinant flu vaccine works over 40% better at preventing culture-confirmed flu than traditional egg-based flu vaccines. And so the focus of our platform is both really a speed and safety benefits, as well as now the efficacy benefit. So speed, really, because we can make vaccines much faster using a plug-and-play technology, and the safety because of the purity of the protein.
Thanks very much, guys. So we chose this group of panelists because they represent different stages of development of platforms and products within their platforms. But I think it's worthy to have a brief discussion about how you define a platform. Is it exclusively the expression system, or do you include some of the purification components in the way you think about a platform approach?
So I can start out with that. So our platform is primarily expression system based, although we have investigated the use of different fusion partners and tags as a method for purification, so that's definitely an advantage as well. But in our case, the main thing would be the protein expression system. Marrying the two actually would definitely speed things up a bit for specific programs. Yeah.
So I would say from our perspective, the main focus of the platform is really the expression system, so plug and play. So what we do is take whatever antigen that we want to express and program the baculovirus. We make a recombinant baculovirus to express specifically that antigen. But besides that, it's the same master baculovirus that we use for all of our products and the same universal cell line that we use for everything, which now is FDA approved. And that really speeds things up from both a production and regulatory standpoint. But from the downstream side, I would add that for our flu program, we do have a platform really for purification, and we have what we call a universal production or downstream platform, because the challenge of a seasonal flu vaccine is that the antigens change every year. And so that the only way logistically to really be able to handle that is to have an approved downstream process where we use the same major downstream purification steps, so the same filters, same resins, et cetera, to get pure protein, but we are allowed a minimal amount of freedom to make minor adjustments to that process. But that really aids in being able to produce an updated product every year in a timely fashion.
I guess finally, in the case of Protalix, again, since we're focusing on using plant cell suspension culture, it's primarily the expression host. But in our case, because we actually had to pave a new pathway to figure out how to scale up and industrialize suspension cell culture for plants, we actually had developed some proprietary bioreactors design. So part of our platform is, and what's novel and proprietary about it, is then the FlexBag bioreactor system that we then designed and developed to enable robust, scalable production of the therapeutic proteins from our